So laurel wilt disease, new invasive threat to sassafras trees. So here's a couple pictures of some symptoms of that. So what you might be seeing are dead trees along the side of the road. Maybe they've been dead for a little while, maybe a year or two, and you haven't really noticed them. Or maybe they just died more recently or showing some weird symptoms. And then cutting into the bark, seeing what's causing those problems. So that's the fungus, that's the laurel wilt disease fungus, the black staining, streaky staining that you see in that trunk, cutting off the circulation of the tree. So let's back up a second and talk about, the background of this disease. So laurel wilt disease, I'm calling it of sassafras here because that's the species that's impacted in our area, has actually been in the United States for some time. So it was first detected in 2002 in Georgia, in this area. And you can see the colored counties are the counties where it's known to be. And the different colors are the year in which it was first detected. So you can see it's in this area for a while. And there it's not on sassafras, it's on red bay laurel, which is a common tree in that coastal area. That's what gives it its name, laurel wilt disease, red bay laurel. It can attack plants in the Laureraceae family. So that's the laurel wilt side of things. In our area, it's just going to be sassafras and spicebush. Although its name is laurel wilt disease, it does not impact mountain laurel or most of the trees that we have. And it's really unlikely to shift hosts. I know people always have questions about that, but it's going to be impacting sassafras and spicebush. And you can see that for most of its time, it kind of stayed here on the coast, in this coastal area, spreading little by little, maybe a few jumps over here. And then last summer, we had this big jump into southwestern Kentucky, northwestern Tennessee, and then a tiny little pocket here in East Tennessee. Obviously, that's concerning for us because this kind of jump like this wouldn't have happened naturally, right? There's going to be some human movement that would result in laurel wilt getting from one of these counties over here. Not that it can't move on its own, but I think a really good example of how we can accidentally move things around. And that's just where we know it is right now. It might be kind of more widespread than that. So what is this disease? It's caused by a fungus that's carried by an ambrosia beetle, the red bay ambrosia beetle. Now that's a very tiny, tiny little insect, and I'll show you some pictures of that. And it can move this around of its own accord. So it's not like the fungus is going to stay put, but really humans moving it around, I think long distance is the bigger challenge. And then both of these are invasive. They're not from here, not from this region. They are from Asia. And I believe Dr. Dreaden's going to talk a little bit more about that in his presentation. But just to note that they're not native to this area. Our trees don't have defenses to them. If we do have a lot of native ambrosia beetles, and we have native fungi that they carry. So ambrosia beetles will carry these fungi with them. They're fungus farmers, and they'll actually eat the fungus. We have that, it's pretty common, but this is a different scenario because while those native ambrosia beetles are typically attracted to stressed trees, trees that maybe are already on their way out, this one is attracted to healthy trees. And the fungus, instead of just causing a little bit of damage, like most of those ambrosia beetle associated fungi, can kill the tree. So a big difference there. And here's where it is right now that we know of, again, in this area. So let's talk a little bit about these hosts. So in addition, I mentioned sassafras. Spicebush is another host that can be killed by this disease. And I've got a couple pictures of spicebush, just in case you aren't familiar with it. It's a tree that grows in the understory. I've talked to foresters and landowners who don't like it and get frustrated with it because it can grow so densely. But it's a fantastic plant to use as a native alternative to a lot of invasives. It stays kind of small, like a shrub. but it's got great foliage. And if you can't see here, the leaves are kind of folded over here, and you might wonder kind of what's going on there. It's also the host plant for the spicebush swallowtail. So these are caterpillars of that swallowtail, and these are some of the beautiful butterflies, the spicebush swallowtail. It also produces these fruit that are good for wildlife. So a lot of benefits of spicebush. And then this is sassafras. Now, I love sassafras as a tree. It's got these really distinctive leaves, the spoon, fork, and mitten. Some leaves are just these single leaves here, but then some have these deep lobes. They might have 2 or 3 lobes. So they can be really different. They smell fantastic. They can grow into small trees, but frequently, or large trees. So some of the areas that we visited that have laurel wilt right now are huge. And the world's largest sassafras is actually in Annette's County, and it's really, really large. But most of the time when we think of sassafras, we're thinking of trees that are maybe in the understory, maybe along fence rows. Sassafras is about 1% of all of the trees in Kentucky, and it's not going to be a major timber species, but it is really important ecologically and just a cool tree. It's also what they use to make gumbo filet, so if you're a fan of Cajun food, you can think about sassafras for those reasons. It's, I think, a beautiful tree in terms of fall leaf color. That's how it looks in the fall, so increasingly popular in some landscape areas. Really nice fall color. People do like using it for wood turning and different very specific woodworking uses because it's got nice wood that can be worked well, but there's just not a ton of it, which is why it hasn't really become a popular or common timber species. But so that's a little bit about spicebush and sassafras, the two plants that are being impacted by laurel wilt. But what should you look for if you're thinking that you might have some in your area? So this is kind of a common symptom, a red flag, and it's actually red for potential laurel wilt problems. So this is a sassafras tree, and you can tell by its different leaves, but it's going into what looks like fall leaf color, right? It looks like fall has come early. You have these beautiful deep reds and oranges. But for this tree, it's not because it's getting ready to senesce and it's winter, it's because it's really water stressed. So the name laurel wilt comes from the fact that the fungus and then the plant's defensive responses to that fungus clog up the vascular system of the tree and basically cut off that flow of water up to those leaves. Eventually that will kill the tree, but in this time window, what you're seeing is signs of water stress. It looks like the tree is water stressed. You might notice a really distinctive leaf color. That can be a sign of laurel wilt. And then if you look a little closer and you were to chip off the bark on the trunk of a tree that has those symptoms, what you might see is this dark black staining just under the bark. Very streaky, very dark, almost a black color. And it is a destructive sampling, right? It's because you're cutting away the bark. And you know, it can be something that if you're not sure that that's what's wrong, you don't want to do that to your tree because that would negatively impact the health of the tree otherwise. But let's say you do this and you find these kinds of symptoms. To me, that's a really, that's really likely to be laurel wilt. There are some other things that can cause kind of similar looking issues, other vascular problems, but the streaking pattern of laurel wilt seems to be pretty distinctive, very dark in color. And another thing is if you have kind of smaller plants, this is on a large tree, but if you were to have some twigs or smaller shoots that have those symptoms and then cut that, you might instead see the streaking just around the outside edge. So you can kind of see this black color. Other fungi that cause vascular problems might also have streaking, but I think this one's darker and more distinctive. So I mentioned that it's spread from tree to tree by these very small red bay ambrosia beetles. And when I say small, I mean super small. So here's a picture of me holding a chip that has that black streaking, but it also has some holes from these beetles. And you can see they're tiny and they are really easy to miss. So I think it's unlikely that you're going to notice the beetle holes or the beetles themselves. What you're going to see are the symptoms in the tree. But some interesting things about laurel wilt. If you're familiar with some other vascular diseases of trees and their interactions with insects that move them around, some background on laurel wilt. So laurel wilt, the damage is really being done by the fungus. Just one introduction of that fungus into the tree is enough to kill it. So you don't need huge populations of these red bay ambrosia beetles to kill the tree. This is different from some other diseases or ambrosia beetles in general, where you're going to see a tree that all of a sudden is just covered with ambrosia beetle damage and tiny little toothpicks that are sticking out of the tree. With laurel wilt, some of the places that we visited, it can be hard to even find some of those beetle holes, but nonetheless, the tree is dying and has laurel wilt. Those beetles can move the fungus from tree to tree. Also, the fungus is vascular, right? So it can move around in the vascular systems of those trees, which is really significant when it comes to sassafras, because sassafras is frequently clonal. That means the root systems of those trees are going to be connected, and the fungus could move from tree to tree in the root systems, especially if you've got some shoots that are coming off of roots. You can see it progress that way as well. So while I really want to mention these beetles and the role that they play in moving around the disease, you might easily miss them. If you're looking for them, you might see these tiny little toothpicks of frass that they make. So that's kind of a combination of the wood as they're chewing through there and their excrement that comes out in these little toothpicks. Now, because these beetles are so small, that can be hard to miss, but something to look for. And then this is about the size of them, right? So this is a tiny little vial that I could carry in my hand, and you can see these little red bay ambrosia beetles, just to give you an idea of that. Now, there are many different similar beetles that appear on sassafras. And as you know, it's not unusual for sassafras to be stressed and declining. So these are some different beetles. So this is a hole caused by the red bay ambrosia beetle, the one that can carry laurel wilt. And this is a different ambrosia beetle. So just even on a tree that has laurel wilt, there might be other beetle damage. So if you see something like that, it's not necessarily, a sign that your tree has laurel wilt. Here's another tree that I found, the sassafras, that when I first saw it, I thought, oh, this must be laurel wilt. But then I looked, and those toothpicks and those holes were just slightly too large. So when sassafras trees start to decline, they are really attractive to ambrosia beetles. So you might have something else. And if that happens, it's a sign that that tree is on a downward spiral and is probably not going to recover. But just wanted to throw that out there, that it's not necessarily laurel wilt. But if you do have laurel wilt, unfortunately, there's no management option available, although there is some research currently in progress. So if you've got a site in an area with laurel wilt and want to be a part of that research, again, we'd love to have you. So it's testing fungicide efficacy for protecting large, healthy trees. There's not going to likely be a way to retroactively cure a tree that has it, but you might be able to protect those trees with fungicide treatment. What is key is limiting the movement of infected wood, because while those beetles can certainly move around and move it of their own accord, we don't want to move it longer distances than that. And it would be really easy to accidentally move around some contaminated firewood from one site to another and move laurel wilt that way. So I think sharing that message is very important. Avoiding transport of wood products, of firewood, of anything that might be contaminated with laurel wilt. And so here's something that I put in there, remove and destroy infected trees. Now, this might be beneficial if this is something you can do and have the resources for. For a lot of people, that's going to be impossible to remove and destroy all of their infected trees, cut them down and put them through a chipper. However, if you have other sassafras on your property that you want to protect, that's really at this point the best alternative management treatment that we've got. would be to cut down and chip the trees that are infected, because that will kill the ambrosia beetles and prevent them from moving into your healthy trees. It's not a perfect solution, it's not a great one, but that's what's available right now. And even if you do that, there's no telling that it wouldn't move in from another area or already be there and you can't see it, but it is something. And then I put this in here, of course, you all are mostly county agents, so you're well aware of this, but encourage people to contact their office if they're seeing anything like that. Myself and the Division of Forestry Specialist, Alexander Blevins, are also happy to work with people if they have questions about that. So with that, I'm going to turn things over to Dr. Dreaden, but I want to mention that we have a new laurel wilt disease fact sheet out that myself, Dr. Dreaden, Jonathan Larson, Kim Leonberger, and Nicole Gauthier worked on. You can find it if you go to forestry.ca.uky.edu/laurel wilt. And then we also provide information on Instagram and Facebook related to forest health topics. So with that, I'm going to stop sharing and see if you all have any questions for me before I get Dr. Dreaden on. All right, well, let me know if you do. Tyler, feel free to go ahead and share your slides if you'd like, or maybe introduce yourself and your background with laurel wilt a little bit.
So as Ellen said, I'm Dr. Tyler Dreaden. So I'm with the USDA Forest Service Southern Research Station, but I'm located here in Lexington, Kentucky. I'm part of the Forest Health Research and Education Center. But I did my my graduate and postdoc work in Florida. I worked on laurel wilt disease there in the lab I was in. Did a lot of laurel wilt research. So thank you, Ellen.
No problem.
So I'm just going to give a short introduction to some laurel wilt disease research. And then the image you see there on the right is just an example of what the pathogen Rafflesia luricola looks like in culture. Next slide, please. So as Ellen said, laurel wilt disease has many different components, but I'm just going to talk about some current and past research on the three main components. So the pathogen, Rafflesia luricola, the vector, the red bay ambrosia beetle, Xyloborus scobratus, and the host, which is the Lauraceae family. A few examples of those are, sassafras, avocado, and red bay. When you get all of those combined, you can get laurel wilt disease. Next slide. So the vector and the pathogen are native to Southeast Asia. But as Ellen said, in its native range, they're not really studied very much because they don't cause disease. They act like typical ambrosia beetles, and they're attracted to dead and dying trees where they make their galleries and eat their ambrosial fungi, and don't really cause a problem. But they're introduced into the Southeast US around 2002, and they act completely different here. Next slide. So this is a typical disease cycle for laurel wilt disease. So in their native range, the red bay ambrosia beetle are attracted to dead and dying Lauraceous hosts, like most ambrosia beetles. So they bore into these dead and dying trees, make their galleries, grow their ambrosial fungi, and both the adults and the next generation eat the fungi, and then they emerge and start the cycle again. But again, in their native range, they're attracted to dead and dying hosts. But here in the US, in the introduced range, they act very different. So the adult red bay ambrosia beetles are attracted to healthy red bay trees. So this is illustrated at the top of the screen in the disease cycle. Yes. So the beetles bore into these healthy trees to initiate a gallery, but they soon abandon these galleries. And it's thought because they find the trees to be healthy and not suitable for brood production. But as they initiate these galleries, they inoculate the pathogen into the vascular system, and wilt starts to develop within a few weeks, and the trees wilt and are completely dead within several weeks, at which point they become very attractive to the beetles, which come back and start their gallery constructions. Then the adult and the next generation feed on the fungi in these galleries. The next generation develops and repeats the cycle, infecting and killing healthy hosts. So again, in the native range, the red bay ambrosia beetle act like typical ambrosia beetles and infect dead and dying trees. But here in the US, there's some sort of a mismatch, and they're attracted to live hosts. And the pathogen can kill large trees within a matter of weeks. Next slide. So Ellen's already shown this, but the beetle was first detected near Savannah, Georgia in 2002, and it spread throughout the southeast. Try clicking. But there's been several jumps in the distribution of the disease. So here's an example where the disease jumped to Jacksonville. Click again. And there's another advance there in the peninsula of Florida. In this case, we know what happened. There's a wood turner that visited Jacksonville, Florida, and found this dead red bay tree with that really dark, beautiful stained sapwood, and thought it would make some amazing bowls. So he brought the bolts back to his shop, and several months later, all the red bay trees in his yard were dead. So we know this jump in the distribution was caused by people moving infested material. But there's been several other jumps. Ellen, do you mind advancing until the Tennessee-Kentucky circle shows up? There we go, thank you. So there's been several other jumps in the distribution of the disease, and this has led to many questions. Do you mind advancing again and again? So how many introductions? Is this just one introduction into the US and then people moving infested material around like we found with the wood turner in South Florida? Or are these new introductions from Asia into the US? And does the isolates that are found in the US, do they represent more than one clone? Was there multiple individuals introduced in the US at one point? Or are there multiple individuals introduced into the US at multiple points in time? For sexual reproduction to occur, you have to have two mating types. So we wanted to know if both mating types are present in the US. We also want to know, what the source of the US population was. Next slide. So this is a study we conducted. I won't get into the details, but this is a population genetic study using molecular markers. I just want to highlight a few things in the diagram. So isolates in the US are in the purple color, and the lines connecting different circles, the thicker, the darker the lines, the more closely related those individuals are. And the mating type is indicated by the color of font. So isolates from the US were found to be in 3 different genotypes, and they're all very closely related with a single mutation and a single location separating the 3 different genotypes. And they're all in black font, so they're all the MAT2 mating type. Yes. We also recovered 1 isolate from Taiwan that was, yes, the same genotype as the majority of all isolates we found from the US. We found much more diversity among isolates from Asia and both mating types. Now we found 3 clusters. One of all the isolates from the US, Japan, and Taiwan, and the isolates from Myanmar were quite different and formed 2 separate clusters. So these results highlight we need to limit future introductions of the pathogen into the US, because right now there's a very limited diversity, and we need to continue to monitor the isolates in the US and incorporate any new genotypes into a resistance screening effort, which we'll go into a bit more detail later. Next slide. So a little bit more about the genetics of the system. Concerning the red bay ambrosia beetle, Xyloborus glabratus, it was recently split into 3 taxa, and because of this split, it's unclear about exactly where each of these new taxa are distributed globally, but only 1 genotype was found in the US, and the same genotype was found in Taiwan. And for the pathogen, there's limited diversity within the US and only a single mating type. So right now, sexual reproduction in the US is probably not happening. And the same genotype found in the US was also found in Taiwan. So Taiwan's a possible origin of the US introduction. And because of the limited diversity of both the vector and the pathogen, it's likely a single introduction of both of them at a single time into the US. And this highlights that we need to prevent future introductions. Next slide. So maybe an appropriate title for this disease or talk would be Attack of the Clones. But since both the vector and its symbiont are clonal, I like to call it the attack of the symbioclones. Next slide. So we're going to shift gears and talk about host resistance for a minute. And this work was conducted at the Forest Pathology Lab at the University of Florida by Dr. Jason Smith and Mark Hughes. And this is just an example of a large red bay tree there on the right. And they can get fairly large when they hit enough site resources. Next slide. So this is some of Mark Hughes' research and slides. So he went to stands that had a high density of red bay trees and where these sites were just devastated, where mortality reached almost 100%. So the top left image is an example of one of these stands where there's a high density of red bay and virtually all the trees have died. So he would survey these impacted sites and try and find large, surviving, healthy red bay. So right there, the tree in the center is one surviving red bay that is surrounded by a dead red bay. So we take cuttings and then propagate these in a mist bench, the photo at the top right, and grow them in a nursery for use in resistant screening work. Next slide. Unfortunately, small trees give inconsistent results when screened with a pathogen. So we then planted these trees out in the field so they could get much bigger before doing field inoculations. So this is an example of one of his field plots where he planted selections that survived in the field in multiple replicates. So we inoculated these trees by drilling a small hole at the base of the stems, so a single inoculation site, and pipetting in a spore suspension. Next slide. So he would evaluate the trees 12 weeks after inoculations. If you look at the right, this is Mark Hughes by one of his trees. You can see they need to be a decent size before the screening results are consistent. And it takes quite a while from going from a cutting from the field to getting a tree in the ground this large. So it takes time. But the vast majority of the trees looked like the one on the right. So they completely wilted and died by 12 weeks post-inoculation. But there were a few clones, like the one on the left, that showed some tolerance, where just a portion of the crown would wilt, or a single branch, but the tree would survive and continue to grow. They actually went back the following year and re-inoculated these surviving trees, and they got the same result, where a portion of the crown would wilt and die, but the trees continued to survive and grow. So this is very promising that they can find some host resistance. Next slide. So some of the next steps in this research, they want to identify more tolerant individuals and then start breeding these tolerant clones together to try and increase the resistance. They're also working to develop a restoration strategy and a plan for deploying tolerant individuals. And then, as Ellen mentioned, the Forest Service in UK is starting a study to look for tolerance in sassafras. And hopefully we'll have some results in the coming years. But trying to screen for resistance, it does highlight the need to limit future introductions of both the pathogen and vector. Because right now there's a single clone in the US of both the vector and the pathogen, which makes screening for resistance easy because you only have to use a single isolate. If we get more diversity in the pathogen in the US, we'll have to start screening individuals with multiple isolates. And also, new introductions of the pathogen could break down the resistance that's already been identified. So it really highlights the need to limit future introductions. Next slide. So a little bit on management. As Ellen said, fungicide injections have been studied, and they have been shown to be effective in avocado, red bay, and the research on sassafras is ongoing. But the trees need to be re-injected every year or two. This is also quite expensive and really is only practical on high value trees. It's not even economical in avocado orchards as a preventative strategy for all the trees. Sanitation has been tried in native stands of red bay where it was not very effective, but in avocado orchards, it has been effective and is recommended. So the photo on the top right is an avocado orchard, and the dead trees are laurel wilt. In avocado orchards, the disease progresses a little different than in red bay, where root graft transmissions are significant. So in the bottom right photo is the same orchard when all the diseased trees were removed, following strict sanitation measures. And this has been shown to be effective in avocado if sanitation is applied early and completely. And like I said, resistance has been identified and is being pursued as a management strategy. And people are hopeful that one day it will be a realistic option. Next slide. There's also some work on controlling the vector. So different chemicals in the host were identified to both attract the red bay ambrosia beetle and be a repellent. So this is some fairly new research and it's ongoing, but they're hopeful that they can develop a push-pull strategy as part of a management strategy in avocado orchards. And it might be deployed to other systems. So this is pretty exciting, but ongoing research. Next slide. So in the beginning, there was hope that cold winter temperatures could control the northern limit of the disease because the vector is sensitive to cold temperatures. So using some laboratory work, Formby et al. Developed a model to see how winter temperature might control the vector populations. Unfortunately, they found that there wouldn't be much of an effect as far north as us here in Kentucky. And some of the lab results were confirmed when we found the infestation in Kentucky and Tennessee. But some ongoing research should help tell us, how the winter temperatures here will affect the vector. But we're not as hopeful as we once were that the winter would significantly slow it down. Next slide. So there's a recent review of Laurel wilt and natural systems published in Forest. I encourage you to look at it if you have more questions or reach out to me. And that should be it.
Great. Well, thank you so much for all of that fantastic information. For everyone who's joining, Tyler is really steeped in the work that's ongoing with Laurel wilt, both kind of where it came from and now what do we do about it in our forests. So if you have any questions for him or me, be happy to take those now.
So there is a question on the origins of the isolates that we find here in the US. Unfortunately, the vector and the pathogen are native to a large area in Southeast Asia, and we only have samples from Japan, Taiwan, and Burma. So it's hard to say exactly where the US introduction came from, but we did find the same genotypes of both the vector and the pathogen in Taiwan. So given the very limited sampling and limited information, our best guess is it came to the US on imports from Taiwan, but this is very preliminary and just our best guess at the moment.
All right, we have a question from Susan Fox. Will the fungus move into other saplings connected to the infected tree if it is removed? Also, do they always bore into the trunk or also the scaffold branches?
So yes, it can move through the root system. but it doesn't seem to move at least amazingly well through the root system in avocado. It has been shown to move through the root system in sassafras, but I guess we'll see how well it does here.
And with does it move into the trunk or the branches? The beetles are attracted to the bigger, kind of the bigger trunks first, but it doesn't, from my observations in sassafras, seem to preclude the smaller trees from becoming hosts as well. And I assume that the same would be true about the branches, but Tyler, do you have any info on that?
Yes, so as you mentioned, the beetles are first attracted to larger silhouettes, but they will eventually bore into the smaller branches too.
Yeah, Lori, I agree. I also really hope it doesn't wipe out our avocado trees or our native sassafras. And I think it's really exciting that people are finding resistance. I think of, when these invasive insects and diseases have come in, we have a few different examples of what they've done to our native trees. So you've got kind of the American chestnut scenario where really we're not finding native resistance to chestnut blight. You still find some trees, or actually they're more like shrubs these days, out in the woods, but it's not that they have any resistance. It's just that they're able to grow enough before they're killed by the pathogen that those root systems can stay alive for some period of time. So that's kind of one scenario. Whereas on the other hand, you've got something like Dutch elm disease, which wiped out huge numbers of elms, but there were about maybe 10% of the trees that had some resistance to Dutch elm disease. So you still find elms around. There's a lot of variation in resistance in different elm species. So a different scenario. With emerald ash borer, we're also finding that there are really, really, really low levels of resistance, natural resistance. So out of entire states, there might be a handful of trees that have some genetic resistance that make them poor hosts for the emerald ash borer. And we don't really know what the story is going to be with sassafras yet, and I think the jury is still out on that. But at least there does appear to be some resistance in other Lauraceae trees to this disease, which I think is really promising. Any other questions, or have you all been seeing this kind of thing in your area? Oh, your sassafras sapling appears to be infected. Susan, is this I can't remember, did you send me a photo of that? And I need to get back to you on that. Yes, so if I recall, it was a small sapling in a field and there were no other trees around it. Is that right? Like a small tree, and there weren't kind of other larger sassafras that had those symptoms. So I will say this, in that it is not at all uncommon, and those of you who are taking calls from the public on this know, to get reports of sassafras trees that are declining. There's a number of different things that can stress and kill sassafras, not to mention the fact that a lot of where they tend to grow, these roadsides, old fence rows in the understory where they can be outcompeted and shaded, those aren't great from its health and growth perspective. So there's a lot of things that can impact sassafras, and it's not uncommon to see some in decline. This past year, I've seen a whole bunch of death of newly planted seedlings because of, I think, our weather conditions over the past bit. So we had that extreme drought last summer. If newly planted trees were not watered, there's a really good chance that they would not survive because they didn't have established root systems. And then we had the last fall where trees did not go into dormancy before we had that frost hit, that can stress trees. We had those 2 late frosts this year. I know that many trees, and I can't comment on sassafras, but put out a flush of leaves and then they were fried, and then they put out another flush of leaves and they were fried. And I certainly had some seedlings die from that. So there are other things that might be killing your sassafras, but that aren't laurel wilt in addition. But if you do think you have laurel wilt, maybe this is a good time to talk about what you do about that. So let me go ahead and share my slides once more.
Ellen, that tree's been looking fine all summer. It's a sapling that just came up volunteer, but there are some root suckers off of it that look fine.
Mm-hmm.
I know some of the upper leaves on it probably have the butterfly larva on them.
Oh yeah.
But the lower leaves didn't, seem to be look like sassafras wilt, but I haven't scraped the bark yet. I may do it because there's other, suckers coming up that I can go ahead and let grow.
Yeah, and I will say that if it's suckering, I feel like it's unlikely that you're just going to get symptoms on one. You might initially, but then it would spread to the others, would be my thought. So if that's the case, yeah, whenever, if it continues to go downhill and it's kind of small, I would either kind of scrape off the bark in a place and see if you see that black staining, or if you want to cut the whole thing, and then you could take a little segment of it and send that into the lab if you do see some of that black staining. The diagnostics lab have their procedures for submitting samples, and I just wanted to go over those for laurel wilt in particular. And Tyler, feel free to correct me if I say anything wrong on this, or if things have been updated, because I know that you've been working with the diagnostic lab to look at potential laurel wilt samples. So feel free to update me. But if you're looking at a mature tree, and this is submitting a sample to our diagnostic lab to see whether or not it is laurel wilt on a mature tree, so a large tree, it's tricky because what you're doing is destructive to the tree, right? So you do want to make sure that there's something wrong with that tree before you start hacking into it. But on the main trunk, use a hatchet or a hammer or knife or something. And I've had success putting like a hatchet in there and kind of hammering it in a little bit to remove the bark down to the surface of the sapwood. And it's not always in that outermost layer, but usually if you go around it, a tree that has a lot of staining and you're in an area where laurel wilt might be, look under the bark, and if you can find that stained wood, that really dark streaking, cut out a few thick chips with that staining, and that's what you want to submit. If you're looking at kind of smaller stems or saplings, you can use a knife to kind of shave back the bark and look for staining just under it. And if you see some, then you can cut and collect that whole section, like just using some pruners or something. And for both of those, don't send dead samples. So if it's already dead, they're not going to be able to culture the fungus necessarily out of it. And that's going to be necessary for confirming whether or not it's laurel wilt. Put those in a plastic bag, keep that cool, and get that to the Lexington lab as soon as possible. And Tyler, correct me if I'm wrong, but I think that the Lexington lab is still the one that's handling all of the laurel wilt disease samples. Is that correct?
Yes, as far as I know.
Yeah, so get those to the lab there with Julie Beal and Sarah Long, and that will initiate that process of trying to figure out what it is. And I will also throw in there, Tyler, feel free to share anything that you have on that, because I know you've been dealing with this for a while. The fungus, Rafflesia lauricola, grows really slowly. So if you're wondering like, oh, I submitted these samples forever ago, why haven't I heard back from them? It's in part because at this point, the test that's involved in this involves culturing the fungus from that wood and then sequencing those isolates. So it's an involved process, but to kind of make it worse is that fungus grows really slowly. So it can take a while to determine whether or not it is positive. Is there anything else you wanted to share on that with people if they're prepping samples to submit?
Just try and get them as fresh as possible. So, you know, as the tree is wilting, is best. If they get too far gone, you can get all sorts of other fungi. And yes, with sassafras, it seems to be much harder to isolate the pathogen, and you get all sorts of other fungi.
So fortunate that we've got such great diagnosticians working on this in our state. So that's wonderful work that they're doing, but it can be a little challenging. So that might be why things take a little longer. Any other questions? All right. Well, if that's it, I just want to thank all of you for joining today. I want to thank you, Tyler, for joining and sharing your work with us. Really interesting and exciting potential future solutions to this problem. Thanks all of you for joining today and being with us. And if you have any questions, feel free to email me, e.crocker@uky.edu, and I can pass them on or direct them. So thanks for coming.
And thank you for the opportunity.